Let's move on to our next speaker, our final speaker of this set. And so we have Jean Macklaim, a bioinformatics scientist at DNA Genetech. She'll be talking today about compositional data analysis approaches to improve microbiome studies from collection to conclusions. So I'll turn it over to you.
Hi, can everyone hear me? I apologize. I don't think I can get my camera working, but I hope you can see my slides and at least hear me.
Yes, yeah, I can hear you fine. And let's hope some of the participants can too.
Let's hope so. So thank you for having me here. I'm really excited to be here, at least virtually. And especially thank you to the organizers for putting on this day. And, you know, in spite of our pandemic world and the challenges we're facing, we're able to talk a little bit about microbiome data. So as a broad overview for my talk today, I'm going to be covering what compositional data analysis is and why does it apply to microbiome analysis in particular. I'm going to give some examples of applications that we've used this compositional data framework in our internal R&D efforts, and I'll go over some general concepts that are applicable and useful for biological discovery and prediction. So in order to generate microbiome sequence data, there are a number of steps your samples have to go through. So you start with a total bacterial population that you are sampling in some way. And then of that sampled portion, you're extracting either DNA or RNA in the sample. And then you may be amplifying or producing a sequence library out of that. And at the end of the day, at the end of your sample process, you are collecting reads from a sequencer. So there's two key takeaways from this process. The first one is that we are subsampling a very small portion of the original starting material. And the other point is that at every step of collection, processing, sequencing, and analysis, there's potential to add unwanted bias or unwanted noise into the data at the endpoint. So there's a recent paper from Macklaim et al. That outlines the result of this problem, where your input sequence or your input profile of microbiota that you're trying to measure going through this process may not represent what you intend to see. So I'll give a small example to illustrate the problem. So here I have a microbiome profile, a very simple one of 5 taxa. And the challenge is trying to figure out or trying to determine which taxa are changing from A to B to C. So for example, if we look at this orange taxon in the middle, it looks like it's increasing in abundance from each of these conditions. And then conversely, we can look at this purple taxa at top, and it looks like it's decreasing over those conditions. But if I show you the underlying data, what's actually happening is that orange taxon from A to B is unchanging, while from A and B to C, it's a 6-fold change in proportion. The purple taxon that I mentioned as well, from A to the final point at C is actually unchanging, while the middle point is decreasing. So the problem here is proportions. The problem occurs because we can't actually measure the underlying population from easily or without challenges or cost. And so if we represent as proportions, every component influences another component that we're measuring. So this is a compositional problem. So sequencing data are therefore compositional. Anything that can be represented as part of a whole or part of a total is compositional. And as I mentioned, the change in one of those components can affect every other component. So by sequencing, you're subsampling. So it's as if you're reaching into a jar of Skittles and grabbing a handful, but you're not grabbing every Skittle out of that jar. The counts you get back from the sequencer are unreliable. So if you take the exact same sample and you sequence it again, you're not going to get the exact same counts represented in your seq data. And this is especially prominent for rare features that have the highest uncertainty. Proportions, so out of say 100%, cause problems. And that's because the relationship between those components are distorted from the truth. And in representing your counts data as proportions, you're also losing information about the library size and the variation of the data. And ultimately, you're breaking the laws of statistics and being able to analyze your data with traditional methods. A zero is not necessarily zero. So microbiome data are sparse and variation comes from the low counts. There are true zeros, so taxa say that's in one condition and not another, but those are often indistinguishable from the normal sample variation or undersample problem that we just can't capture those rare taxa. And the biggest problem is that common normalization and statistical methods are not well suited for these type of data. They can't account for that variability, the sparsity, and the fact that the data doesn't fit normal assumptions of data analysis. So how can we approach these data and overcome the challenges that I mentioned? So we've developed a compositional analysis workflow shown here, which has equivalent tools and strategies to a standard or a common approach in a microbiome analysis study. So I'm going to highlight 3 key features from this toolbox, and I'll be showing examples today to demonstrate this. So the first key feature is the normalization step. So in a compositional framework, rather than trying to normalize the data are transformed into what's called log ratio space. So this means that the ratio of the parts is conserved and the parts are no longer dependent on the other parts of the composition. So that's fixing the problem I mentioned earlier about proportions influencing components. And in addition to that, I'll mention there's a bit of a secret sauce applied where the CLR transformation is combined with another estimator of technical variation to account for the variation problem I mentioned. The second part is after transformation, using some kind of distance metric and ordination representation to determine what similarity or differences are in the whole microbiome. So in compositional space, we're applying an Atchison distance and a compositional biplot. And then the third point I'll mention is that after transforming the data, the next question is, can we evaluate the individual changes between samples or between cohorts? So in this case, the per sample univariate comparisons are done with a compositionally aware tool like ALDEX. So for the rest of the talk, I'm going to demonstrate these in this framework applied to our ongoing R&D projects. So we're applying these across a variety of challenging sample types, working to improve all aspects of these studies from the collection to conclusions, and showing that these applications extend to any kind of microbiome sequencing study trying to uncover biological inference. So the first example I'll highlight is from low biomass samples. So in particular, we're interested in developing methods and tools to collect, to sequence, and to analyze these sample types, such as skin and urine. So in particular, low biomass samples are challenging for a number of reasons. There's very low DNA input. It's very hard to extract enough content. They tend to have high host DNA contribution as well. And because of these factors, the data are very sparse. They contain lots of zero counts, and they're highly variable and less reproducible. So perfect candidates for a compositional problem. So this particular data set example I'm going to show, we had collected samples, midstream urine samples from healthy male and female donors, and the samples were held for 14 days in a stabilization chemistry before shotgun sequencing. And the objective here was to confirm that an individual's baseline microbiome profile could be determined from that endpoint sample. So this first representation I'm showing is using a traditional non-compositional distance metric with the sample distances represented in a cluster diagram. So each point is a different sample here. So most samples, as expected, pair, meaning baseline and endpoint are the same or similar in microbiome composition, but there are a number of samples that do not co-cluster, marked in red on the diagram. And in particular, I'll point out female 9 here, where the endpoint and the baseline do not co-cluster in this methodology. I'll also note that, importantly, the common normalization techniques that are often applied do not fix the problem. And even worse, they can lead to sample loss due to low read counts and higher false discovery rates for downstream analyses. In contrast, here's the same data using compositional distance metrics. So this is the Atchison distance of the CLR transform data, and with that technical estimator I mentioned before. And now in every single case, each pair of samples co-clusters, so baseline and endpoint together. The additional benefit is that there's now an obvious cluster group here aligned with the environmental control samples. So we know that these samples are likely impacted by background contamination and noise compared to the other samples. And if we look at the 16S copy number of that sample group measured by qPCR compared to the rest of the sample set, they are on the extreme low end. This is about 400 copies compared to the rest of the data set, which is closer to 500,000 16S copies. So I'm going to pull out female 9 as an example here of what the profile looks like at baseline and at the endpoint in proportional space. And the problem is exactly the one I described initially in my introduction example, in that this large shift of lactobacillus, this blue organism, is driving the proportional difference between these two samples if you look at it in a non-compositional way. In contrast, when this is transformed into log ratio space, as I showed before, that bias is removed because the relationship between each ratio of these parts is conserved. So this is well described. There's the paper I mentioned, Macklaim et al., where they walk through in silico and mock community examples that demonstrate this property, that proportions don't necessarily give you the right answer, and that log ratio data preserves the expected profile. So the next example I'm going to draw on is one about impact of method choice. So choices that you make upstream in your sample processing that could later impact your data. So the data here are stool from healthy adult donors, and every donor had 3 extraction replicates, and they were sequenced with metagenomic shotgun sequencing. So I'm showing 2 broad methods of extraction, and in each case I have the non-compositional on the left, the compositional on the right, and showing the difference between these methodologies in the extraction replicates versus the between donor difference that we expect. So the problem here in a non-compositional space is that the distance is constrained. It has to sum to 1. And so it's obscuring the variation that's underlying between these methodologies. In contrast, using the adjacent distance, there's a higher resolution to detect the changes, and there's more information content. And in this case, we can truly see that there's additional variation in method B, adding noise to the extraction replicates compared to method A. And so this is an application that allows us to choose a best method based on trying to reduce the amount of bias that you're inputting into your sample upstream of the data analysis. I'm going to jump now to a more complex data type using metatranscriptomics. So broadly, beyond taxonomic assignment, metatranscriptomics tries to address what the functional contributions are of the community. The challenges here in the data types are extensive. All the way from collection, RNA is not very stable. It degrades in minutes, and it's hard to capture an accurate RNA profile. And we have similar challenges to low biomass samples where there's high sparsity, high variability in the data, and this property of asymmetry. And this comes from the fact that we're measuring transcript abundance, but the underlying composition or the DNA can also influence those changes. So here we took stool samples from healthy donors with 2 broad methodologies over different conditions, and compared the total RNA-seq transcript composition and taxonomic composition. So these are represented in compositional PCA biplots. So in blue are the baseline samples that we're comparing to, and in green and red are the 2 methodologies that were included for comparison. So in this case, in the taxonomic representation, it's already apparent that chemistry R, so one of the methodologies, has a profile different from baseline, whereas the green chemistry here is more overlapping with what the expectation is. This is even more pronounced in gene function composition. So here again, the green chemistry is overlapping with baseline, while red is differential. But if we look at the loadings in the background, so these are the gene functions contributing to those changes on the plot, you can clearly see these associated gene functions that are driving those differences away from the other two comparisons. And that's also apparent in the first principal component, where we have more explanatory power by showing that the gene functions are different. I'll contrast again to a non-compositional approach. This is the exact same data represented in Bray-Curtis dissimilarity peak-CoA plots, and the data are colored either by chemistry or by donor. But in this case, the effect of the chemistry is obscured, and I hope you can see that the clusters associate with different donor groups, and you're not actually able to detect that underlying variation that I just showed is actually driving bias between the upstream choice. So I've highlighted a couple examples of how compositional can impact your interpretation of low biomass samples and how it can affect your choice of method. So I'm going to walk through an example of how we can evaluate changes over time. So this data set again were stool samples from 18 donors with replicates held in different storage conditions, and metagenomic shotgun sequencing was performed. So looking at the Atchison distance, our baseline comparison is the replicate variance. So these are just sample replicates in what we expect from any normal sample process. The samples that were stored in OM chemistry are on par with sample variation. And then the gold standard -80 collection is indistinguishable. So these are good methodologies for collection. In contrast, samples held at 60 days at room temperature show a huge shift in the microbiome composition, approaching that of the donor-to-donor differences. So now that we know there are changes in the whole microbiome, how can we test what those changes are? So these are the exact same data evaluated with ALDEX2 for differential expression. And so on each of these plots for conditions, I'm showing the difference between the conditions scaled to the difference within each condition. And this is an effect size. The criteria for determining something that's different is whether anything, as in this plot, exceeds that effect size of 1. So that means that the between condition difference far exceeds the variation within the donors of the condition. And again, each condition shows that there's very little variation, but room temperature at 60 days has a few very specific taxonomic changes that I'll show here. So as an example, I'm showing 2 changes that occur, 2 different taxa that have shifted over time. One has an effect size of 2. This is a log scale, so it's 2 to the 2. This is really apparent that these means are different and they're non-overlapping in their distributions. For an effect size of 1, which is just on the borderline of the cutoff criteria, the relative abundances of these taxa are overlapping. And so if you are trying to determine which biomarkers or which components are most valuable in interpretation, you'd probably look at this effect over this one. These, of course, are both significant by p-value, so by using just p-value, you wouldn't necessarily know the effect of that change. And just as an example here, I have the same data comparing those differences between times with a small sample size and a large sample size, showing that p-values will increase with sample size, whereas the effect size is stable and consistent. Just to wrap up, the benefit of using compositional data analysis is it allows the data to be used for other applications as well. So this was a paper I was involved in. Data were transformed compositionally for microbiome 16S and compared to compositional data from metabolites. And what compositional data allows is that you can apply normal statistics, which we picked out a predictor of change tied to a microbiota function that was then validated in vitro as well. So to summarize, biases in microbiome studies can be introduced at every step from collection, processing, sequencing to the data analysis, which impact your ability to interpret and discover. The compositional analysis framework can effectively identify and mitigate the problems with complex, sparse, and variable microbiome data. Integrating these analyses into our R&D have improved our ability to develop reliable microbiome sampling and sequencing solutions. And if you look at the literature, these compositionally aware analyses are providing insights for biological discovery, hypothesis testing, biomarker discovery, and other multi-omics analytics. So I'll quickly acknowledge, of course, my teams and colleagues at DNA Genotech, Diversigen, CoreBiome, who have been involved and instrumental in some of these data evaluations, and then past and continual collaborations with Dr. Greg Gluer, who developed a lot of these methodologies. And just before I close, I'll mention that you can reach out and connect with us. We have a team of people ready to provide service consultations, free trials of our collection devices, or get involved in many of our collaborations. So if you want to stay involved or reach out, please go to trydnagenotech.com/myproject, and we really hope to hear from you. Thank you, and I can take questions.
Thank you so much. So I think we're just about perfectly on time here, and we may not have another minute for questions because we're going to a break right away, but rest assured, if any of the audience members would like to ask a question to Dr. Macklaim or the team at DNA Genotech, please enter it into the chat function, and then we'll make sure that it gets forwarded to her. So thank you so much, Dr. Macklaim, for your presentation.
Thank you.